Hello there. I'm glad to welcome you at my virtual poster booth. My name is Skorodumov. I'm a PhD student at the Central Institute of Mental Health in Mannheim, Germany. And today I would like to present you our recent study in which we assessed the efficacy of psychedelics in a DSM-5 based rat model of alcohol addiction. So alcohol is one of the few legal psychoactive drugs in the majority of the Western countries. And yet, according to the World Health Organization report from 2018, each year more than 100 million disability-adjusted life years are lost due to problems associated with alcohol misuse. As of today, there are 3 approved medications on the market: disulfiram, naltrexone, and acamprosate. But the success rate of these pharmacotherapies is quite low. And in this regard, data from the early and recent human studies with psychedelics clearly suggest that they have a high potential for becoming a unique approach in psychopharmacology. So the main hypothesis nowadays is that a single or a few sessions of psychedelic-assisted psychotherapy could restore normal brain function and result in long-term positive outcomes across many mental disorders, including addiction. However, in clinical studies, it is very difficult to disentangle the purely biological effects of psychedelics, which apparently involve changes in synaptic plasticity and so on, from those that are driven by the supportive environment of psychotherapy. And therefore, animal models can provide a good opportunity to resolve this dilemma. Now, for a person to be diagnosed with alcohol addiction in the first place, he or she must fulfill certain criteria which are defined in DSM-5. First, the consumption of alcohol must be compulsive, meaning that the person is not able to control urges to drink despite knowledge of negative consequences. And second, this person eventually relapses even after long periods of abstinence. So the model which we use in our lab adheres to these criteria, and I will now quickly explain how it does so. When the rats are 2 months old, they are housed separately in single cages, and in each rat is given free access to 4 different bottles: water, 5, 10, and 20% of alcohol. The exact order of the bottles in the cage is changed weekly to minimize habitual preferences which may develop over time. And this drinking phase lasts for 8 weeks initially. After that, alcohol-containing bottles are removed from the cages and animals are put into deprivation for 3 to 4 weeks. At the end of this phase, the alcohol bottles are reintroduced again, and this cycle repeats 2 or 3 more times with different duration of each phase, again to minimize and to prevent the habituation effects. Eventually, those rats will develop a sustained compulsive drinking behavior, and after each deprivation, they will consume significantly more alcohol during the first 3 days than they did during the last week before the deprivation has started. And this is called ADE, or the alcohol deprivation effect. So this model provides accurate phase and construct validity because it mimics the relapse behavior observed in humans, and the compulsive drinking in rats develops voluntarily under a free choice access to alcohol. And this is also why we use 4-bottle paradigm instead of 2 bottles to give our rats more freedom of choice. But most importantly, this model also offers predictive validity because acamprosate and naltrexone, which, as I said, are approved for human use, are similarly effective in this model when tested. And to test the drugs, they are administered at the end of the deprivation phase to see how they would affect the upcoming ADE. And that's how we know whether the particular drug prevents the relapse or not. So we decided to test psychedelics in this model using 3 different dosing schedules. So in the first subset of experiments, we administered 1 milligram per kilogram psilocybin in a model-specific way that is used to assess the pharmacological effects on the relapse behavior in this model. So the first dose was given 12 hours before the reintroduction of alcohol bottles, and the remaining 4 administrations were done across the first 2 days of drinking, with the intervals of 12 hours in between. Second, we administered 8 microdoses of psilocybin over the whole deprivation period of 4 weeks, with gaps of 2-3 days in between of the injections to mimic the human definition of microdosing, because the administered dose was also 1/10 of the full dose, or 0.1 milligram per kilogram. And in the third subset of experiments, we applied medium and high doses of psilocybin and LSD twice, 1 week and 12 hours before the reintroduction of alcohol, to replicate the study design by Michael Bogenschutz and colleagues from 2015, in which they administered psilocybin in 2 separate sessions to patients who were also suffering from alcohol addiction. In each experiment of our study, the drug group was compared with the vehicle receiving group, and all experiments were done in parallel on male and female rats. So we found that psilocybin significantly decreased alcohol intake in rats when it was administered in a model-specific schedule. And this finding clearly speaks for the presence of some biologically mediated mechanism of action, because this effect of psilocybin was slightly stronger than the similar effect observed in the same model under the same administration schedule, but when we administered acamprosate and naltrexone. However, it is clearly obvious that such dosing of psychedelics is not suitable for the human use, because in this case, alcohol intoxication overlaps with the psychedelic experience, and all the human sessions are done when the patient is sober and abstinent from alcohol for at least 3 days. But unfortunately for us, we didn't observe any long-term effects of psychedelics, neither in the microdosing nor in the human-like medium and high dose administration schedules. And this could be explained by 2 different options. The first is that our model itself is not able to capture the different aspects of psychedelic-assisted psychotherapy, and therefore the findings from the human studies cannot be translated into the animal model. Or the other option is that the biological effects of psychedelics alone are not sufficient to reset the impaired neural circuits and normalize the behavior of the addicted individual. Anyway, at the moment, we can say with certainty that only the future studies, which will be using different models of alcohol addiction and different schedules of psychedelic application, will clearly shed more light onto this question. So that was it from my side. Thank you for your attention. I hope I was able to explain you everything. If not, if you still have some questions, please feel free to contact me via this email, and see you all at the next psychedelic conference. Stay safe.